difference between primary literature and secondary literature. Primary literature is a research paper published in a journal, so that's the one we've been reading most of the discussion. You all have free access to the Chris Library for the New York Times. So if you go to the library resources, you can click here for an academic pass, and that usually gives you 1 day access to the New York Times. And then you can click on that, you enter your UNO ID, it authorizes you, you get free access. So you can start looking there. The science section of the New York Times is pretty well organized, and most, if not all of their articles will cite primary literature. They might not be dealing with genetics, so that's the part you have to pay attention to. You don't want one that's necessarily environmental science or science that doesn't do genetics, how we define that kind of reflection. Any questions on that? So your article is due, finding an article, an appropriate article, and posting it is due April 4th, I believe. It should be a month long.
Yeah, can you show me that weekend from the library?
I Googled Chris Library New York Times, and this is what it popped up, but I think you can get there from the library too. We'll see. I'm typing on the wrong keyboard.
If you go under academics,
Yeah, so I think there's a lot of resources here. So I don't know, I just Google it, but I don't know which tab will be in here. But if you have a different periodical that you can't get access to, like the Lancet, the New Yorker, they should all be in here too. So you can search kind of these databases here. But I would just Google directly New York Times, UNO, or Chris Library. Okay, so today we're going to finish translation. So this big section we've done on the central dogma, DNA replication, transcription, translation. Yesterday in discussion, we had practiced doing translation, and now we kind of want to give the context of the extra details that are happening mechanistically. And so the part of translation that I find the most difficult is that you're trying to visualize a three-dimensional process that's happening continuously in kind of these still shot things. So we're going to try to walk through the bits by bits, how we've done with the other processes. There's a lot of different ways that different textbooks represent these structures. I wouldn't get too familiar with any particular structure except highlighting key things, right? There's parts of the ribosome, it's reading the RNA in a specific direction, and there's specific parts of the ribosome, EPA sites, and we'll talk about what those are and what they mean. So our central dogma that we've been working on this whole time, DNA, RNA. Right, the first lecture we did replication. So you saw that yesterday in the discussion. We had that fork, leading, lagging. That's because of polarity of the DNA molecule. Then we saw transcription. And this is really a two-step process, right? It's the process of actually synthesizing that RNA molecule and then processing it, if you're in a eukaryotic cell, to get past that nuclear membrane. And now we have translation. So these were nucleotides. The nucleotides, so in the same language. And now we're translating to a new language, a language of polypeptide, which is composed of amino acids. So how are these connected? The RNA is the template. We've kind of been using template in many different contexts. But you can think of the RNA as the template that the ribosome is reading for translation. And I talked about this in my sections yesterday, but I don't know if Dr. Gruey did, so I just wanted to kind of make this really explicit. The modifications that we were thinking about are only specific to eukaryotic. So in a prokaryotic system, this is what's happening. So you have your DNA, so it's open. It looks like a replication fork. It's not. It's just open more locally at the area that you're interested in. So the green is the DNA. You have RNA being transcribed. And in prokaryotic cells where there's no nuclear membrane, the ribosome, so small subunit, large subunit, can make its polypeptides immediately. There's no intermediate stage. It's immediate translation from your RNA template. So for eukaryotes, we have what? 5' cap, poly A, tail, splicing. All of these were to help export and stabilization. So just in case you didn't think about that in the terms of why these things are functional, now that should be explicit. So how are we going to go in between the two languages, from the RNA nucleotides to amino acids? We're going to use the genetic code. So how many, does anyone remember off the top of their head how many amino acids are?
20?
Yeah. And how many bases do we have? In our RNA sequence, how many possible nucleotides are there? Yeah. So we need to make a code that can take information that has kind of 4 unique properties and translate to 20 unique things, right? So we can't use each individual base to represent an amino acid, right? 4 to the 1 is 4, that won't give us enough. Or we can't do doublets, so that's about 16. We can use triplets. We can make 3 base pairs together that we're going to call code for a single amino acid. This is an excess, right? There's 64 possible codons, right? 4 choose 3, so 4 cubed. And part of this gives what we call the redundancy of the genetic code. Some amino acids are coded by multiple codons. And we'll see that even though that's the case, there's still a lot of specificity in the system that maintains kind of accurate transcription translation connections. So this is what the codon table looks like. So your one for discussion was mislabeled. You can read this a couple ways. The way I like to read it is I start with my kind of the left-hand side and go inward. Then the second letter, and then look within that. And I think this highlights the redundancy of the genetic code, right? If you look at alanine, so down here, the first 2 bases are all the same, G and C. The third base is different, but it has no functional consequence. You're still going to code for alanine. So when we think about mutations in RNA being carried forward into the polypeptide sequence, not all mutations are going to be equivalent, right? So this third position speaks directly to that redundancy, and if mutations occur there, no change to your protein. If you change the first or second, often you'll have a change in your protein because you'll have a new amino acid sequence. And so the other key thing about this table that you should remember, like you don't have to memorize this table, I give it to you, but different tables are not always labeled the same way. So here, that AUG, that's the thymine right here. So I would memorize that one as being your start codon. And then I would memorize these 3 as your stop codon. So in this table, they're labeled stop, they're not always labeled stop. So you only have 4 codons to memorize, the rest you can generate from just using this table. The reason to memorize the start and stop codon, because if you're scanning a nucleic acid and you want to quickly identify a coding sequence from untranslated region, those are kind of the endpoints that we're dealing with here. Any questions on that? So you've gotten practice at this, using this yesterday in discussion. So why these triplets? What do they do functionally? So this is a figure from your book, and it's showing kind of the DNA at the top and then what it would look like in the nucleotide sequence. So again, we have a template strand and a non-template strand. Those are anti-parallel and complementary. The mRNA is going to be anti-parallel and complementary to which strand?
The template?
The template, yeah, and you can tell by the polarity, right? Anti-parallel means they're running in a different direction. The RNA has to be built in a 5' to 3' direction, so that also gives you a hint about who is anti-parallel to who. And then you get this amino acid. So these codons, there's a step that's missing that we're going to talk about, and that's the tRNA in a couple slides. But there's another anti-parallel complementary sequence that gets you from this mRNA to the protein, and those are the tRNAs. So again, polarity is so important to dictating how this process works. So I know you've practiced this already, but these are kind of letting us practice as a class like this, and they help get our top cut out of the way. So what is the RNA transcribed from this sequence of DNA? So this should be in top cut. Yeah, so the answer I'm looking for is like a string of nucleotides. So what's the key thing to remember? Keep answering this, but we're going to start it kind of together. So I'm seeing a couple of different answers, and there's one that's predominating, and we'll see what's happening there. There's another one that differs by the identity of the single base, like one sequence is using uracil, one's using thymine, or thymine. And then there's one that's doing something interesting at the end. But so we're going to be an RNA now. So should we have any T's in our sequence?
No.
No, there's no T's in the codon table either, right? So remember when you're kind of answering a question like this, RNA uses uracil instead of thymine. So the template strand is this bottom strand, and we're going to build an RNA 5' to 3'. We're going to move towards the right. So this is going to be A, U, G, right? That's going to be the first 3 bases that are going to be transcribed. And then you can keep going on. E, C, C, right? You keep going down the list. Some people have kind of picked up on a trick, and if you want to apply this trick, that's fine, but I would really think about the mechanism behind it, that you're going through kind of this template strand directly. Because on an exam, you might be given template, you might be given non-template, you might be given an anti-codon. You should think about how these are all connected. So the trick that some people are using is they just are substituting the U's here, right? What do I mean by that? They're just replacing U's. So it's A, U, G, G, C, C, A, U. So you don't have to do the kind of mental gymnastics of, okay, what's complementary to T? It's an A. You can kind of look at that non-template strand. But if you're doing that, think about what's actually happening. So the best answer here is just AUG, GCC, AAU, GAC, UUU, CAA, UAA, right? Just replacing that non-template strand with uracil. So what's the protein sequence going to look like? So I'm going to write the RNA again while you're working on the protein, just so we can read it. how I use this table is, you want triplets, right? You want to identify 3. And this question, maybe you don't have enough information. We'll talk about reading frame in a little bit. We talked about it in the discussion yesterday. Usually you don't want to just assume that you're starting in a specific reading frame. So reading frame means how you're identifying triplets, right? I could identify triplets like this. I could identify triplets like this too. So these are alternative reading frames. Both of them have an AUG, a start codon. But we'll see in a couple slides, it's the first AUG that you encounter that's going to dictate your reading frame. That AUG, something happens specifically when a ribosome encounters an AUG before translation starts. So we're going to use the black reading frame instead of the blue. The blue is a good alternative. You just end up with an AUG later on. So if the problem does not tell you the reading frame, do not just assume you start kind of chunking it up in 3, so you have to find that first AUG. We're going to use the black reading frame. So AUG, they come here, right? A, U, and I look into it, it's methionine. It is the start codon, so it's good that a lot of you are recognizing that, but I wouldn't put start, right? Start is not an amino acid. It's not gonna, it's good that you're recognizing that, but I would put methionine. Methionine is the amino acid that corresponds to that codon. The first one, the first methionine is the start codon. So we can do it again, GCC, alanine, right? And so on and so forth. Any questions on using this codon table? You end up hitting a stop codon. I see people putting a stop in.
No.
What's that? If you entered a stop codon, what stop codon did you hit? TAA. So UAA. Yeah. So that's your stop codon. So that's UAA. If you look that up on the table, UAA stop codon. So you can put stop. There is no physical amino acid that's going to be incorporated, or you can just stop the sequence. It's up to you and how you want to be explicit about it. But I like putting stops because sometimes we'll see a mutation can introduce a stop where we might not expect one. So I think putting stop explicitly is important. But if there was a stop, don't keep going. The stop is, it's like a hard stop in this transcriptional process and translational process. So any questions on using this codon table? Yeah.
This is the happen that we have like a double AUG in the same strand. Can we like even though we are crossed up, can we stop by the new coding?
So I think I'm going to try to repeat your question. So you're saying there's another AUG here, right? Is that what you're picking up on this one?
Yeah, after the stop.
After the stop codon?
Yeah, let's after the stop you are cross again AUG.
I think that could occur, but I don't think, so after a stop codon, so we'll see what happens in translation, but after there's a stop codon, the ribosome actually disassembles and comes off the RNA. So that RNA will then be degraded. So I think it's very unlikely that you'll kind of start again. The ribosome would more preferentially, because the ribosome is always going to start from the 5' side of the RNA, it's going to hit the first start codon again. So things can happen. Things can jump past stop codons, but I think that's pretty rare, unless some genes are transcribed in certain ways that we're not going to talk about. But yeah, if there's another start codon, I would not think about it. For the purposes of this class, we'll consider like a stop, a hard stop. And you'll see the ribosome physically comes up off the DNA. And if it were to restart, it would start back at 5'. It wouldn't start kind of where it left off. That's a good question. Thank you. So what's ensuring the specificity between RNA and DNA? There's really 2 important steps. First is the specificity between the tRNA and the amino acid. Okay, there's a physical link between these two molecules, and this is facilitated by an enzyme. And so ASE tells us it's an enzyme. What is this enzyme doing? It's synthesizing a bond between tRNAs and amino acids. So it's amino acyl tRNA synthase, but that's its job. It's making a bond, a physical connection between a tRNA and an amino acid. So the tRNA has an anticodon that's going to come into the ribosome and interact with the RNA. That's our first level of specificity. Our second level of specificity is the correct match between the anticodon that's on our tRNA and our codon. So one of these is a covalent bond, one of them is a hydrogen bond that's taking place within inside the ribosome. So some tRNAs can bind at multiple codons, given that third base pair kind of has some wobble. We're not going to consider that. We're going to try to really focus on accuracy on both of these two steps. So if there's multiple codons that code for the same amino acid, we'll see it's not the same tRNA. It's individual tRNAs with unique anticodons. So this is what's actually happening. This is one representation of what's happening inside the ribosome with this tRNA anticodon and the codon interacting with the RNA. So this is not just happening free floating. This is within the ribosome, but that's kind of not shown. So you can just focus on what's happening. So that tRNA has that clover leaf that holds on itself. It has an amino acid, so here's the thymine, and then you have that anti-parallel complementary anticodon. So this is to highlight that an individual tRNA binds to one codon. So we've seen a couple representations of tRNA before. If you're showing it in kind of a two-dimensional, it's because you really are paying attention to these hydrogen bonds and the different arms, and we're not going to pay attention to that. We really only care about the anticodon and the amino acid attachment. So how is the amino acid attached to the tRNA? So these are not from your book. I don't think your book had very good figures about this. So what this is showing is the little green squares are individual amino acids. The tRNAs are kind of the little loops I've shown you before, all with their anticodons. And then there's a physical protein, this amino acyl tRNA synthase, that has kind of binding sites, and it's catalyzing the reaction. So once they're together, it's now a charged tRNA. Charged and uncharged don't really matter for our class, but what matters is that each of the tRNA and amino acid only fit in one amino acyl tRNA synthase. So there's not 64 of these proteins, there's less because of stop codon, but there's way more amino acyl tRNA synthetases than there are amino acids. It's the same number of tRNAs. So that's where that one-to-one relationship and specificity becomes big. So this interacts with one tRNA and one amino acid. So that's what I'm trying to show by the different shapes. Like they only can fit in certain configurations. So here's an anticodon. Don't pay attention to the sign, the little ends of the modified bases. So here's an anticodon, CGU. Which amino acid is attached to this tRNA? So think about all the anti-parallel complementary sequences, and remember that this is an mRNA codon table. So people can answer, but we can talk about, where are you gonna start if you have a problem like this? What's your first step going to be? So I think there's a lot of people getting the two most common answers. So for people who answered this, how do you go about answering this question? Yeah.
You look at the directions of the anti-codon.
Yeah, I think that's a good place to start. So what does looking at the direction do for you? For me, it helps me identify the codon. So if this anticodon on the left-hand side is 3', the right-hand side is 5', there must be a codon in this orientation. Okay? The codon will be antiparallel and complementary. So what is the codon we're dealing with? What's the sequence, nucleotide sequence of our codon? Yeah, it's complementary. GCA. If this table is for codons, you can still learn something about the amino acids through the anticodon, but you have to apply the table at the codon level. So here's GCA, down here with an alanine. So if you put arginine, it's because you just read the anticodon, right? You went CGU. So the best answer here is alanine. You have to recognize that antiparallel complementary nature between the anticodon and the codon. Any questions on this? So I think I had a slide on this, yeah. Just showing explicitly kind of the relationship between the codon and anticodon. So now we're going to think about translation. You've been thinking about it, the components, you need this tRNA charge, specificities from this aminoacyl tRNA synthetase. We have anticodons, let's look at them in the context now of this ribosome. So this is what we're building towards, right? Ribosome with subunits moving from 5' to 3', RNA sequence, all of these charged amino acids. So this is happening kind of continuously. And we're going to just try to break down the different steps of these spectrums. So I'm going to use figures that are not in your book, because I like them a little bit better, but I'll try to also show you the figures in your book. So if you're familiar with the figures in your book, you can kind of make these connections. So these are the basic parts of our ribosome. So we have a large subunit, a small subunit, and these are composed of both RNAs and proteins. So it's not shown here, it's just this big yellow blob, but there's a lot of different RNA and proteins compacted into the structure. Proteins, why? Because proteins are necessary to carry out reactions, they're catalysts, they have catalytic sites. But some of the RNA have catalytic sites as well. Why do we need catalysts? Why do we need enzymes? Because we're going to be transferring, we're going to be breaking one covalent bond and forming another covalent bond as we grow this polypeptide chain. So the proteins here are enzymes. But both the RNA and these enzymes have catalytic activity. And then there's 3 sites labeled EPA, Environmental Protection Agency. So what is the, does anyone remember from the reading what these sites are? You can start left to right. So the first site that's occupied is actually the A site. So there is an order, and that's really important. So a lot of people call them different things. So A, you can call it the aminoacyl site. You can also call it the accepting site. I don't really care as long as you're recognizing that that's where these tRNAs are initially entering the ribosome. So P is the polypeptide site. kind of where this polypeptide is gonna start growing. And then E is the exit site. So it has an order. Right? It's moving to the right. If this was a 5' end of an RNA, it would be moving in this direction. So RNAs are going to come to the A, P, E, and then exit. And it's not like they kind of sit there. We're going to see snapshots, but this is really rapid. They're just kind of transiently passing through this ribosome. So this is the figure from your book. So they call it the aminoacyl tRNA binding site. So accepting site, amino acid site, peptide site. They're really being specific about what's being connected in this peptide. It's a peptide bond that's happening from one tRNA amino acid to the next, and we'll see that in the exit site. And then so the C is showing it with kind of tRNAs embedded, right? These sites are, after you start, they're unoccupied until that first start codon, but then after you start, they're always three are kind of being occupied and just transitioning tRNAs through them. So that's what this one on the right, the C, is trying to show. So here's some just other diagrams that try to bring that three-dimensional shape. So you don't have to, whichever way helps you think about them, you just have to know their function. And we'll walk through that. So both in DNA replication and in transcription, we have three main steps, right? What are our three main steps?
Initiation, elongation, and termination.
Yeah, thank you. So we've seen these repeatedly. That helps me organize my thoughts, like how are they the same, how are they different? In transcription and translation, we have polymerases for elongation, now we have the ribosome, right? In transcription and translation, we had either origins of replication or promoters. It was DNA protein interaction. We still see like an RNA protein interaction for the initiation, but we have some key components, right? We have start codon. We have the tRNA that's going to have that initial methionine, and then the ribosome, small subunit of the ribosome. And then termination, transcription was terminated. Just in eukaryotes where kind of bubbles met each other. In RNA, we had a terminator sequence. Here we have a sequence that's specific for termination. We have a stop codon. And then we have release factors. So there still are proteins in this process. They're not necessarily recognizing the sequence, but they're really important in catalyzing the reaction. So here's initiation. So what do we see here? The little yellow blob is the small subunit. It kind of has this initial start codon or anticodon, the UAC methionine, being held onto it by these initiation factors. So this is why translation starts at the first AUG. So this little thing here, this little yellow thing would be your untranslated region, part of it. It's actually going to be up until the start codon. Here's our start codon. The ribosome in this configuration can't carry out translation. It's missing the large subunit that has all the catalytic properties. But this is the unit that's going to first attach to the 5' end of that RNA and kind of cruise along until it finds the first start codon. So it has everything it needs to recognize the start codon. It has that first tRNA with an anti-parallel complementary sequence to that UH. So this is why you don't really see translation go past the stop codon, because every time you have to start 5' in this configuration. Any questions on this? When does the large subunit come in? It doesn't come in until contact is made with the start codon. So this is a figure from Khan Academy. It's kind of cartoonish, but hopefully it helps you think about reading frames. I've said that word a couple times. Reading frames are how you are defining your triplets, your three bases that you move down the line. So for this for this RNA sequence up there, there's three different reading frames, right? So the three different reading frames, it's not going to start at it does the one on the bottom, number 3 is the most correct, which starts with the thymine. But if you started in one of the other reading frames, you get proteins that don't really make sense, right? So here you have methionine, and you have a premature stop codon. Premature being that it's internal to the peptide sequence. Here there's no methionine. Here there's a methionine and a correct stop codon. So how is it chosen? It's not chosen like the ribosome's not choosing. If we go back to this slide, there's only one way for it to find the start codon, through these initiation factors and the tRNA. But you're not a ribosome, so how you should go about solving these problems is finding the first AUG. So you find this, and then this dictates the reading frame. So then you can start slicing up your sequence into codons of 3. Any questions on that? It'd be easier if you were a ribosome, right? You wouldn't be in this class if you were a ribosome. Okay, so we've seen this a couple times, but just really to emphasize, the mRNA is read in this direction. Polarity matters even for this level of translation. So let's think about establishing our reading frame. So if translation begins with the first start codon, what's the second codon?
CAC.
I think most of you are getting it. I'll leave it open as we talk about it. So you're a ribosome. You have the small subunit, your tRNA, you're cruising, so you find the first AUG. So, not an A, not an A, not an A. Yep, there's an A. AUG. First codon, start codon. Now I can chunk this up. Second codon, third codon, so forth. So if the question is asking the second codon, it's CAC. And what's really important is that, remember, the codons are in the polarity of the mRNA. So adding these polarities, if you were to write an answer that was just CAC, I'd accept it. But if you want to be really, really explicit, always write your polarities. You can't. More information in terms of polarity is always a good thing, because then I know kind of how you thought about the problem. Or you can explain to other people how you thought about the problem. So the best answer here is CAC. So the ribosome found the start codon. The initiation factors, so these purple things are initiation factors, are released because physically the large subunit comes off. So physically there'd be no room for the large subunit with those initiation factors held in place. So this is kind of the only exception where you don't come to that A site first. You're kind of already sitting on that P site. That's kind of a misnomer just because there never was an A site for this tRNA, right? It was already attached to that small subunit. So for all subsequent tRNAs, they're always going to enter this A site first. So the amino acyl site, the accepting site. It's open, right? There's nothing that occupying it. So there are proteins that facilitate this. We're not going to talk about them. Your book kind of draws it that just, it looks like it's a stochastic process and the tRNA comes in. That's not true. But it is kind of stochastic. It's not like directed. It's not like all of the tRNAs are lining up waiting. They don't know what ribosome they're going to interact with. They don't know what mRNA they're going to interact with. The proteins are kind of bringing it. And sometimes a wrong tRNA will come in, and then because it has no complementary sequence, it will go out. But this process is so fast that often the right tRNA comes in really quickly. So this is elongation. So the next tRNA is going to come into this A site. Some things to pay attention to. This polypeptide has a free amide group here. So that would be the N terminus. It's not drawn here, but there'd be a carboxyl group down here. So that's a C terminus. This one would also have an N and a C. So we haven't talked about those functional groups, we're not going to. I just want to highlight that there is a polarity to proteins as well. As DNA and RNA have our 5' to 3' polarity, proteins have an N to C polarity. The only way to make a polypeptide, a peptide bond, is by connecting a free C terminus, a carboxyl group, to the amide group. So that's what happens here. So what changed? This is the polypeptide that's going to be built. But this tRNA is going to eventually need to leave the P site and go to the E site. So the polypeptide sequence has to change, right? So it was, in the previous slide, it was right here, right? It was connected. Now there's been a physical change. The proteins and RNA within the ribosome facilitated the breaking of the one peptide bond, or not a peptide bond, the bond between the tRNA and the amino acid, and now the creation of this new peptide bond. So this red is that peptide bond. So there's been a physical transfer of this growing peptide to the new tRNA. So for people who might care about the side groups, those are kind of what they look like. Or sorry, the functional groups, the amino, the amine groups, and the carboxyl groups. And now it keeps moving down the RNA sequence. So now this was our initial. This was kind of the anticodon for the start codon. It's now outside the ribosome. It went through the E and kind of popped out. We have this new tRNA that was once at the A site is now at the P site, and the A site is open again. So this just keeps repeating over and over and over, so you still hit a stop codon. Any questions on that? This is fast. It's not like it kind of clumps along like a machine. It's like a 1/10 of a second that every amino acid is being incorporated. You can build proteins really, really quickly. So this is the thing from your book. So it shows its tRNAs and those little blue things. What it's trying to show is the dynamic system of them kind of filtering through different sites. So what are you responsible for at this level? I think, given a picture, you should be able to identify where you're at. Are you at initiation? Are you at elongation? Are you at termination? Which comes first, right? If there's an empty A site, so empty A site here, that comes first, and then you have the peptide bond, right? So this would be number 2. They're labeled. I'm just trying to draw a picture. Then you have the transfer of the polypeptide. And then you have the exit. So if you saw a picture like this, and it might not be arranged like this, hopefully you'll be able to identify where you are in this process, like which came first. Any questions on that? So at some point, that cycle continues till termination. So termination occurs when there's a stop codon is reached. There is no tRNA that codes for a stop codon. Instead, there's a kind of a protein that we call a release factor. So this occupies the A site. So you still have that polypeptide sitting there at the P site. There's no tRNAs coming. There's no tRNA that codes for stop codon or release factor. I think there's a picture. So the discovery of this release factor has won this person a Nobel Prize. He was actually a professor at the University of Utah, where I was before coming here. His name's Mario Capecchi, and he has a road on campus named after him. So he discovered release factors. So this is what it looks like. So this is from your book, right? It's not, it looks like the tRNA in the sense that it has that weird little shape how they're making it in your book. But the key thing is there's no anticodon and there's no amino acid. right? The peptide is done, it stops. So they're showing like it kind of fits within there, but right, they're not showing the nucleotides. I don't know if you can see that. So in your book, these little things are the nucleotides. That release factor doesn't have nucleotides. It has shape that helps it fit into that A site, but it doesn't form hydrogen bonds with the mRNA. And so then the ribosome disassociates. So it's not going to go past the stop codon, very unlikely, because now it's not even a full functional unit. Right? So that's what's shown on the far right-hand side. That mRNA is now most likely going to be degraded. It's very rare that multiple proteins are translated from one mRNA. So like we saw with other, mostly I guess just RNA, proteins, the polypeptide isn't a protein till it's been processed. So we're not going to talk about it a lot, but there are other enzymes or other protein chaperones that then, you take this kind of string on a bead and you give it a three-dimensional shape. So you're kind of going here from like a secondary structure, a tertiary, or even a quaternary structure if you end up getting multiple dimers in together. So I think we're going to be done really quickly. I only have a couple more slides. Any questions on everything we covered? So this, you got practice thinking about the consequences of the discussion. We're not doing that today. Instead, we're giving you kind of the background of what's actually behind the scenes when you're kind of doing these problems. So any questions before we wrap up? So these are the types of questions that you were dealing with a little bit, depending on whose section you were. There's an amino acid sequence, and now there's a mutation. What's the consequence of this mutation? So the original would have been, so I guess the new one here is GAC. So let's assume, do you have enough information to answer this question?
No.
So what are we missing? We're missing the start codon that's going to establish a reading frame. Yeah, so let's just assume we're in reading frame. Let's just assume that's our reading frame. What's going to happen? You can look at your codon table, but I think, personally, answering this question, it might be easier by this process of elimination. I don't have to look at the codon table. So what answers can be eliminated? And shout out one that we want to eliminate. Why? Does anyone want to eliminate unchanged?
Yeah, because you change the T to a G.
Yeah, so if you didn't hear that, usually the identity of the amino acid is determined by the first two bases of the codon. The third is where we get that redundancy. We're changing the first. So it's very likely that we're going to get a new amino acid. So we can rule that one out. I think someone was saying
That was me.
over here.
Yeah.
Okay. What about shortened by an amino acid? Okay. What about shortened by several amino acids? What would be required for it to be shortened by either one amino acid or several amino acids?
It'd be a stop codon, you'd have to start with U.
Yeah, so I heard a bunch of things. I think this one would be through a stop codon. This didn't create a stop codon. So what about B? Someone over here was saying something? Yeah, this would be caused by a deletion. The deletion, if it's a deletion of 3 bases, it's not going to interrupt the reading frame. It will just change the amino acid sequence or shorten it. If it's a deletion by 1 or 2 bases, it's actually going to change your reading frame. So it'll have potentially a really big impact on the rest of your coding sequence. So we got rid of those. Okay, D and E, different after the T to G change. That's kind of a throwaway. I think the best here is different by one acid. And you can actually show that by looking at your codon table. Does a single amino acid change matter? What's the consequence of changing a single amino acid? It can be a lot, depending on where in the polypeptide sequence that occurs. So this is an example from sickle cell anemia. So this is in the beta globin gene. So you see the first codon on the left-hand side, that's for glutamine. Glutamic acid is polar and acidic, right? So it'll be in certain parts of the three-dimensional shape of the protein. If you have a single change here, so we went from GAG to GUG, so this is the mutation, right? That we care about. Now you get valine, which is non-polar. Non-polar thing is another term for that's hydrophobic, right? So if it was on the outside of a protein, now it's going to try to change the conformation of the protein, but also interaction with other proteins. Non-polar hydrophobic things like to aggregate. So now you have a different phenotype, right? You have a very different phenotype for your blood cells due to just one nucleotide change, one amino acid change. So these have really large consequences, potentially. Okay, next one. So the question says the coding strand had a deletion of these three highlighted nucleotides. What's going to happen? And let's assume we're still at 5' 3'. And let's assume that this is still our reading frame. So what's going to happen? So the answers are, some of them are the same, some of them are different. But we can apply the same logic. So it's not going to be unchanged. It's going to be different, looks like a throwaway. So how are we going to disentangle the answers for these other three options we have? So on the other slide, we have this would be a deletion. This would be a stop codon. What about this last one, E? How would you get no product produced? It might not be completely relevant to this example, but just hypothetically, how would you make a mutation that now you never get translation at all? Yeah, yes, I don't know. You delete the start codon. Or mutate it. You don't have to delete it. We're looking for an AUG, and now we only have GUG. There's no other ions. It's very unlikely, but maybe that happens if you delete the start codon. This isn't the start codon. I haven't given you the start codon, but since I've given you a reading frame, you just assume the start codon's upstream. So that gets rid of that one. This didn't create a stop codon. It's just a deletion in frame. So the key here is deletion in frame. Right? Preserves the frame. The frame is the same before and after the deletion. So the best answer here is B. Do we see examples of this? Yes, all the time. So in the beginning of class, especially when we were thinking about pedigrees, we talked a little bit about cystic fibrosis. If you have changes in the length of your amino acid, and that protein is really important for kind of like a domain, like spanning structures, length and size matter, and now you've made a change, you can disrupt that. You've kind of changed the neighborhood that this other amino acids are going to be occurring in. So you can actually make a non-functional protein if this happens. And there's a lot of different, what's really interesting, like sickle cell anemia, it's really most people who have the trait have similar mutations. Cystic fibrosis is not necessarily true. So what is this? We got cut off a little bit, but we haven't talked about the difference between missense and nonsense mutations, but we have talked about deletions and kind of frame shifts a little bit. This is kind of like a histogram. So different positions along the protein, the type of mutation, and how frequent they are. It's not, everyone with cystic fibrosis doesn't have a shared mutation. There are so many different ways that you can change the amino acid length in cystic fibrosis and result in the trait of cystic fibrosis. Okay, so that's all I had. It's really early, so I'm happy to answer questions as a group, or if you want to come. The study guide is up. Hopefully that'll be helpful in you thinking about what you're responsible for and being prepared to do. So have a good spring break.
What do I do for like 9 hours?